The pig, sus scrofa, is a long-time standard model organism for demonstrating mammalian organogenesis. It is agriculturally important and accessible worldwide, which makes it available in sufficient numbers in the laboratory. An added advantage is that compared to smaller laboratory mammalian models such as the mouse, the sectioned pig embryo provides a relatively straightforward orientation, making it easy to identify structures and navigate through organ systems during tracing of actions. Because early developmental porcine stages are strikingly equivalent to comparable stages of human embryos, many important embryological studies have used the pig as a significant research model to study human birth defects. Thus, studying porcine embryology is a window to understanding human development. With the advent of xenografts, such as when a pig heart valve is transplanted into a defective human heart to repair it, understanding porcine organogenesis is essential. Comparative genome analyses can be used to analyze diseases of pigs that relate to humans. Earlier chapters in this atlas have addressed stages of mammalian development up to neurulation. Chapter 12 focuses on pig embryos at selective stages that share an overall similarity to 72 to 96-hour chick embryos and early-stage human embryos. Consider that while selective embryonic stages are demonstrated in this atlas, development is a dynamic process. This chapter assumes familiarity with terms in chapters 9 to 11. Pig embryos are traditionally identified according to their crown rump length rather than their number of somates or days of development, Table A.11. The rhone refers to the highest point of the head, usually above the mesencephalon, and the rump is the lowest point on the trunk. As described in Chapter 2, embryos are typically studied as whole mounts or sections. Due to their thickness, intact pig embryos to be used for whole mounts are first stained and cleared prior to mounting on slides, so that the epidermis and organs are relatively transparent. This allows visualization of both external and internal structures. Care must be taken not to crush these thick whole mounts, and they are best viewed with a stereo microscope. Slides of serially sectioned embryos are D_alicate and extreme care must be taken not to put any pressure on the cover slip. Damage to one slide in a serial section series ruins the entire series. As with chick embryos, Chapter 11, a highly successful approach is to first understand the orientation of structures in whole mounts, before studying corresponding serial sections. A reliable strategy to use when working with sections involves tracing structures for one organ system while temporarily ignoring others, and then going back to trace components of a different organ system. It is helpful to consider that the angle at which each embryo is cut during sectioning is not identical in all embryos, nor is the extent of flexion. Thus, when working with a serial slide series, study the cranial structures of a section separately from its caudal structures. External morphology. Overview. The gestation period of the pig is typically 114 days, 16 weeks. This chapter examines the 6 mm embryo, 19 days, and the 10 mm embryo, 20 to 21 days, in detail, see Table AI1. During this time, the embryo undergoes extensive changes in which differentiation, morphogenesis, and organogenesis form the embryo's tissues and organs. The embryo's body is elongated and linear at first, 15 days. Due to torsion at about 16 days 4 mm, the embryo becomes twisted on itself, Fig 12.1. This torsion is transitory and not visible by about 6 mm, Fig 12.3. Flexion causes the embryo to become C-shaped, 17 to 18 days, -5 mm. Fig 12.2. Flexion occurs essentially in the entire body, and specific regions of flexion include cranial flexure, a ventral bend at the level of the mesencephalon. Cervical flexure, a ventral bend at the level of the myelencephalon. Pontine flexure, a ventral bulge of the hindbrain. Dorsal flexure, a ventral bend in the mid-body region that makes the back convex. Lumbosacral flexure, a ventral bend in the mid to lower trunk area. Tail flexure, a bend in the tail. The tail may be bent ventrally or dorsally. External surface. The body of the pig embryo is divided into head, trunk, and tail regions. The 4.5 mm pig embryo appears elongated, with a large head, Figs 12.1, 12.2. The head has prominences, bulges, formed by the developing eyes, optic vesicles, pharyngeal arches, and pharyngeal grooves. In cleared hole mounts, the skin is transparent enough to see the brain regions underneath, and both otic vesicles, future inner ear, and eyes. By 7 mm, the olfactory pits, future external nares, are visible at the level of the telencephalon, Fig 12.8. The trunk of the 4.5 mm pig embryo also has several prominences produced by the heart, liver, mesonephros, embryonic kidney, and limb buds, Figs 12.1, 12.2. Emanating from the ventral region of the embryo's trunk is the umbilical cord, belly stalk. A slight depression between the heart and liver prominences marks the location of the septum trnns/chrysum that will develop into part of the adult diaphragm. The septum transversum separates the thoracic and abdominal cavities. By the 10 mm stage, the liver prominence occupies a large area of the cranial and ventral abdominal surface, Fig 12.10. The foreleg buds form at the level of the heart, and the hind leg buds are found laterally between the umbilical cord and tail. The two pairs of limb buds appear as small, lateral bulges in 4.5 mm embryos, Fig 12.1. They become paddle-shaped by 10 mm, until about 15 to 20 mm when digits form, Fig 12.18. Along the dorsal surface of the trunk of the embryo are the paired, block-like bulges, the somics. The mesonephros, embryonic kidney, also forms a lateral bulge on each side of the embryo. The pig mesonephros is very large compared to that of chick or human embryos. Porcine mesonephric kidneys must function early in development to remove nitrogenous waste since the placenta is inefficient. In 10 mm embryos, a small ridge called the mammary ridge is located on each dorsolateral side between the foreleg and hind leg buds. It will develop into the mammary glands in both females and males. In 20 mm embryos, nipples are visible along the mammary ridge. A ventral swelling caudal to the hind leg buds is the genitive 11 eminence, genital tubercle, that develops into external genitalia. The embryo's caudal end terminates in the tail. Extraembryonic membranes. These function in protection, nutrition, respiration, and excretion. The pig embryo develops 4 sets of extraembryonic membranes. Yolk sac. Although mammalian embryos do not have yolk, they develop a large yolk sac complete with Vicklin blood vessels, also called omphallomesenteric blood vessels, Figs 12.1, 12.4, 12.7 r. 12.8 to 12.11, 12.15 w. The yolk sac is connected to the embryonic midgut via the yolk stalk, yolk duct, and extends into the umbilical cord. Early in development, the yolk sac produces primitive blood cells and functions in respiration while the placenta develops. Eventually the yolk sac degenerates, becoming a shriveled sac in the umbilical cord. Amnion, thin extraembryonic membrane made of somatopleur, Figs 12.1, 12.10, 12.11, 12.15. Completely surrounding the embryo in a fluid-filled sac, the amnion forms a space called the amniotic cavity that is filled with amniotic fluid. The amnion is attached to the embryo at the umbilical cord. The amnion lines the chorion. The amnion functions in a protective role, keeping the embryo moist. During slide preparation, the amnion may have been removed. Allantois, a diverticulum of the hindgut that expands to a large vesicle connected to the hindgut by a thin stalk called the allantoic stalk. The allantois develops a rich blood supply that includes the umbilical blood vessels, allantoic blood vessels. In 4 mm embryos, The allantois appears as a large semilunar sac connected to the embryo through the allantoic stalk located in the umbilical cord. The allantois functions in gas exchange and eliminates nitrogenous wastes. The allantoic vesicle is removed during slide preparation, but the allantoic stalk is usually present. In 10 mm embryos, the allantoic stalk is flanked on either side by umbilical arteries, Figs 12.15, kk, 12.19. Chorion, an extraembryonic membrane derived from somatopleur. It is the outermost membrane and surrounds the entire embryo, amnion, and allantois. Derived from trophoblast cells of the blastocyst, the chorion develops into the embryonic portion of the placenta. The placenta has respiratory, nutritive, excretory, endocrine, and immune functions. The chorion is usually removed during specimen preparation. Internal morphology. The internal anatomy of the pig embryo is more complex than the chick embryo, however, there are general similarities that make study of the pig embryo less daunting. It is best to fully understand relationships of structures in the whole mount before proceeding to sections. Nervous system. The central nervous system, CNS, is a hollow neural tube at the embryonic stages covered in this atlas. The walls of the CNS have differentiated into three regions that are readily distinguished in transverse sections, Figs 12.7m, 12.15m. The innermost layer, called the ependymal layer, ventricular layer, consists of cells with darkly staining cell nuclei that line the lumen of the neural tube. The middle layer is the mantle layer, intermediate zone. It is densely packed with cells and appears dark due to the many cell nuclei. The outer layer, called the marginal layer, marginal zone, lacks cell nuclei and appears light pink. Lying immediately outside the marginal layer of the brain and spinal cord is the pia mater, which is the delicate inner layer of meninges, membranes, that is derived from neural crest cells and mesenchyme. The pia mater is just forming in 10 mm pig embryos, Fig 12.15c. Cranial flexure has caused the brain to curl on itself so that it appears somewhat hook-shaped in whole mounts and sagittal sections, Figs 9.6, 12.1 to 12.5. The first sections in a transverse serial section series pass through the hindbrain and or rhinid brain, Figs 12.7a, 12.15a. The general locations of the brain regions are shown in Fig 12.1-2c. Telencephalon. The most rostral region of the brain, Fig 9.2, is the telencephalon. In 6 mm embryos, it is a single swelling with a cavity called the tchlococcal. In sagittal sections, it appears near the heart due to flexion, Fig 12.4. At the 10 mm stage, the telencephalon has dilated dorso-laterally to form two cerebral vesicles, future cerebral hemispheres, that are connected to the medial, unexpanded portion of the telencephalon called the lamina criminalis, Fig 12.15p. The cerebral vesicles will continue to expand in older embryos to cover, not only the lamina terminalis, but also the diencephalon and mesencephalon. The external surface of the cerebral vesicles is initially smooth, but will later develop convolutions. The cavity of the lamina terminalis is the median telecele that forms part of the third brain ventricle. The cavities of the cerebral vesicles are the lateral brain ventricles, future brain ventricles I and II. They are connected to the median telecele via the foramina of Monro. A quick way to find the telencephalon in a 10 mm transverse serial section series is to choose sections that are present just after the eyes are no longer visible as one moves deeper into the series. The telencephalon gives rise to the adult cerebrum, olfactory lobes, and corpora striata. Diencephi. On the second brain region is the diencephalon, located between the telencephalon and mesencephalon. Its cavity is the diacelque, which becomes the major part of the third brain ventricle. Although relatively thick in 10 mm embryos, the roof of the diencephalon will thin. Together with the pia mater, a region of the roof will invaginate into the diacele as finger-like projections called the cranial choroid plexum, anterior choroid plexus, that produce some of the cerebrospinal fluid that bathes the brain ventricles and spinal canal. The cranial choroid plexus is visible in 24 mm embryos. The diencephalon also develops several evaginations including the infundibulum, optic vesicles, and epiphysis, future pineal gland. A median diverticulum of the diencephalon floor forms the infundibulum, which is closely associated with an ectodermal outpocketing of the stomodium called Rathke's pouch, Figs 12.5b, 12.7e, 1212c, 12.13c, 12.151l. The infundibulum will form the neurohypophysis, posterior pituitary, while Rathke's pouch develops into the adenohypophysis, anterior pituitary. The optic vesicles are the most prominent ventrolateral outgrowths of the diencephalon of 6 and 10 mm pig embryos. They are connected to the diencephalon via the optic stalks, Figs 12.7g, 12.15m. A depression in the floor of the brain at the level of the optic stalks is called the optic recess. See also the discussion of sensory systems. A striking difference between the 72-hour chick diencephalon and that of the 6 to 10 mm pig is the lack of the epiphysis in pig embryos. The epiphysis forms later in the pig, and is visible by the 35 mm stage as a mid-dorsal evagination of the diencephalon roof. The dorsal region of the lateral walls of the diencephalon will form the adult thalamus. The diencephalon also generates the hypothalamus and epithalamus. Mesencephalon located between the diencephalon and metencephalon, the mesencephalon is the third brain region, midbrain. It is characteristically round in transverse SE sections, Figs 12.7ac, 12.15ae. It is constricted caudally by the isthmus, a narrow brain region between the mesencephalon and metencephalon. The walls of the mesencephalon will greatly thicken as fiber tracts pass through it to and from more rostral brain regions. The mesencephalon floor forms the cerebral peduncles which carry fiber tracts that connect the forebrain the hindbrain and spinal cord. The dorsolateral walls of the mesencephalon form four rounded elevations called the corpora quadrigemina, two superior and two inferior colliculi. The superior colliculi function in visual reflexes, and the inferior colliculi are for auditory reflexes. The thickening of the walls of the mesencephalon narrows the mexicoccal lumen of the mesencephalon, to form the narrow cerebral aqueduct of Sylvius, that connects the third brain ventricle, diaceal, to the fourth brain ventricle, rhombocoel. Metencephalon located between the mesencephalon and isthmus is the metencephalon, which is fourth brain region, Figs 12.7ac, 12.15 AD. Its roof, walls, and floor are relatively thick, which help to distinguish it from the myelencephalon. The lumen of the metencephalon is the metacoccal, which becomes the cranial region of the fourth brain ventricle. The dorsolateral walls grow extensively to form the cerebellum, which functions in coordinating complex muscular movements. Ventral prominences called the pons and cerebellar peduncles form lacer, and carry cerebellar and other fiber traces. Myelencephalon. The most caudal brain region is the myelencephalon, which is located between the metencephalon and spinal cord, Figs 12.L 12.12c, 12.17. The lumen of the myelencephalon is the myclococcal and forms a large region of the fourth brain ventricle. The roof of the myelencephalon is characteristically thin, making it easy to identify, Figs 12.12c, 12.15b. The roof plate and pia mater act together to push inward, ventrally, to form the caudal choroid plexus that produces cerebrospinal fluid. This choroid plexus is visible projecting into the fourth brain ventricle of 15 mm embryos. The internal floor of the myelencephalon is characterized by transient, paired neuromcranes, rhomomers, involved in patterning of the brain, Figs 12.7b, 12. 15 CD. The caudal region of the myelencephalon is flanked by the first paired spinal ganglion called Ferricht's ganglion that has nerve fibers that grow out to the hypoglossal nerve, 12. The myelencephalon forms the adult medulla oblongata. It functions as a reflex center for involuntary activities, i.e., breathing, and a main pathway for connections between the brain and spinal cord. Spinal cord caudal to the myelencephalon is the spinal cord. Although the boundary between the myelencephalon and spinal cord is indistinct in 10 mm embryos, the spinal cord appears smaller, more elliptical, and has thicker walls. It extends into the tail dorsal to the notochord. The dorsal half of the spinal cord is the alar plate, and the ventral half is the basal plate. The eucolimit.ms is a longitudinal depression along the lateral side of the spinal cord that partially separates the alar and basal plates. The roof plate and floor plate of the spinal cord are relatively thin. The ventral region of the mantle layer of the basal plate is thickened to form the motor horn, ventral root, of the spinal cord that sends axons into the spinal nerves. Lateral to the spinal cord are darkly staining, paired spinal ganglia, dorsal root ganglia, Figs 12.7L, 1215G. The thickening of the spinal cord walls has constricted its lumen called the spinal canal, central canal, to become a narrow, diamond-shaped oval, Fig 1215Z. The spinal cord will become surrounded by sclerotome cells, derived from somates, that will form the centrum of the vertebra, Figs 12.7, 12. 13c, 12. 15. Cranial ganglia and nerves. The pig embryo has 12 pairs of cranial nerves that carry sensory, motor, or mixed information, Tabli 9.1, Fig 9.6. They are derived from placodes or neural crest. Cranial ganglia are associated with cranial nerves V, 7, 8, 9, and X. A review of Figure 9.6 will help with slide study of the cranial nerves. Table 9.1 describes their characteristics and function. For clarity, these paired nerves will be discussed as though they were single. Cranial nerve I. In the 10 mm embryo, the olfactory nerve is first visible as faint axonal fibers that arise from the medial wall of the olfactory epithelium, cells that line the olfactory pits, and stretch toward the anteroventral region of the telencephalon wall, Fig 12.15r. See also the discussion of sensory systems. Cranial nerve. Axons from the sensory retina of the optic cup form the fibers of each optic nerve which relays visual information to the brain. In 10 mm pig embryos, axonal fibers of the optic nerve are located in the marginal zone of the wall of the diencephalon, and cannot be identified at this early stage. See also sensory systems. Cranial nerve LLL. In transverse sections of 10 mm embryos, the oculomotor nerve is visible as it emerges from the floor of the mesencephalon to innervate 4 pairs of eye muscles including the inferior oblique, superior rectus, inferior rectus, and medial rectus, Fig 1215df. Cranial NE1T1EN axons of the trochlear NE originate in the floor of the mesencephalon. They grow dorsally within the mesencephalon wall, cross over to the opposite side and exit from the roof of the mesencephalon at the isthmus to innervate the superior oblique eye muscles. The trochlear nerve is not developed enough to see in 10 mm pig embryos, BER is visible at the 15 mm stage. Cranial nerve V nerve fibers of the trich M and .11 neuron, can be observed connecting the floor of the metencephalon to the large semilunar ganglion, Figs 12.2c, 12.3b, 12.5e, 12.7c, 12.8 to 12.10, 12.12, 12.15 The trigeminal nerve is a mixed nerve that has 3 branches, 1, ophthalmic branch, V1 that originated from a placode and connects to the eye region, 2, maxillary branch, V2, that is derived from neural crest and connects to the maxillary process, future upper jaw, and 3, the mandibular branch, V3 that is derived from neural crest and connects to the mandibular process, future lower jaw. The mandibular branch carries motor fibers, whereas all 3 branches carry sensory information from the skin, teeth, and other areas of the face. Of these 3 branches, usually only the mandibular branch is large enough to see in transverse sections of the 10 mm embryo, Fig 12-15 HN. All branches are visible in the 15 mm embryo. Cranial nell VE6. Motor fibers of the abducem neru originate in the floor of the myelencephalon and migrate through the mesenchyme to innervate the lateral recursi muscles that control eye movement. In transverse sections of the 10 mm embryo, the small abducens nerve is visible between the venual myelencephalon and otic vesicles just before both disappear as sections are traced caudally, Fig 12.15g. Cranial neuro 7. The adult facial nerve is a mixed nerve that contains sensory fibers from the taste buds, and motor fibers to muscle derivatives of the hyoid arch, second pharyngeal arch, including muscles of facial expression. Ganglia of cranial nerve 7 and 8 are closely associated as the acoustico-facialis ganglion in 6 mm embryos, Figs. 12.3b, 12. 5e, 12.7c, and become separate in 10 mm embryos, Fig 12.15a. The geniculate ganglion is associated with the facial nerve, and lies outside the myelencephalon, cranial to the otic vesicle and ventral to the acoustic ganglion, Figs 9.6, 12.loc, 12.12d. When transverse sections of 10 mm embryos are traced caudally from the geniculate ganglion, dendrites of the facial nerve can be followed to the hyoid arch, Figs 12.15 fk. Motor axons will develop later. Cranial nerve VLLL lying cranial to the otic vesicle and dorsal to the geniculate ganglion is the acoustic ganglion of the auditory nerve. The auditory nerve carries sensory information from the inner ear to the myelencephalon. In 10 mm embryos, the acoustic ganglion is a single mass, Figs 12.12d, 12.15ef, but will later divide into two separate ganglia each with their own bundle of nerve fibers. The /vestibular ganglion becomes associated with the semicircular canals of the inner ear to function in maintaining balance. Nerve fibers of the spiral ganglion, cochlear ganglion, connect to the cochlea, spiral organ of Corti, of the inner ear to function in hearing. Fibers of the auditory nerve are difficult to identify in 10 mm embryos. Cranial nerve 9. The glossopharyngeal nerve is a mixed nerve that supplies the third pharyngeal arch. Two ganglia are associated with the glossopharyngeal nerve. The superior ganglion is located caudal to the otic vesicle and its sensory roots connect to the myelencephalon, Figs 12.se, 12.7c, 12.loc, 12.15d. The petrosal ganglion is located ventral to the superior ganglion, Fig 12.15g. Fibers of the glossopharyngeal nerve extend ventrally and slightly caudally, and are visible in three regions: 1. Sensory root between the ventral myelencephalon and superior ganglion, 2. Dorsal region between the superior and petrosal ganglia, Fig 12.15e, f, and 3. Ventral region between the petrosal ganglion and derivatives of the third pharyngeal arches. Cranial nerve X. The vagus nerve is a mixed nerve that serves areas of the mouth, pharynx, and viscera. It is associated with two ganglia, Fig 9.6. The jugular ganglion is located caudal to the superior ganglion and lateral to the myelencephalon, Fig 12.15d. The nodose ganglion is ventral to the jugular ganglion and lies along the inner border of the cranial cardinal vein, Fig 12.151l. Fibers of the vagus nerve extend ventrally and slightly caudally and are visible in three regions: 1. Sensory root between the ventral myelencephalon and jugular ganglion, Fig 12.15c. Dorsal region between the jugular and nodose ganglia, Fig 12.15eh, and 3. The ventral region between the nodose ganglion and derivatives of the fourth pharyngeal arch, Fig 12.15rv. Cranial nerve 11. The spinal accessory nerve is a small motor nerve that innervates derivatives of the fourth pharyngeal arch, and certain neck and shoulder muscles including the sternomastoid and trapezius muscles. Axonal roots of the spinal accessory nerve leave the myelencephalon and divide into two groups, a dorsal group that lies next to the dorsolateral myelencephalon and spinal cord, and a ventral group that migrates to the fourth pharyngeal arches, Fig 12.15 cm. Cranial neuro 12. The hypoglossal nerve is a motor nerve that originates in the caudal ventral wall of the mye lens phalon, and emerges as several roots that join and extend caudally to innervate the muscles of the tongue, Fig 12.15 ek. Spinal nerves. The spinal nerves are paired nerves that come from the spinal cord, Fig 12.15 n. Each spinal nerve supplies skeletal muscles from the adjacent myotome of the somite, and receives somatic sensory information from the dermatome of the same somite. Spinal nerves have several components, including a dorsal root that carries sensory, afferent, information to the spinal cord, and a ventral root, motor horn, that carries motor, efferent, information from the spinal cord to its targets, Fig 9.5. Lateral to the spinal cord is the dorsal root ganglion, spinal ganglion, a swelling of the dorsal root of the spinal nerve. Within the dorsal root ganglion are neuronal cell bodies, derived from neural crest, whose axons contribute to the spinal nerve. Between the dorsal root ganglion and dorsolateral region of the spinal cord is the dorsal root, Fig 12.15. The ventral root emerges from the ventrolateral wall of the spinal cord to join the dorsal root, Fig 12.15 m. Each spinal nerve has three branches. The dorsal ramus supplies the epaxial myotome. The ramus communicans, visceral ramus, is a ventral branch that passes medioventrally to the sympathetic ganglion of the autonomic nervous system, Fig 12.15 r. The ventral ramus is a continuation of the spinal nerve that is distal to the junction with the ramus communicans. Because all components of the spinal nerve may not be present in the same section, it may be necessary to view several transverse sections of 10 mm embryos. At the level of the foreleg buds, the spinal nerves form a bundle called the brachia plexus that will give rise to nerves that innervate the forelegs, Fig 12.15 y. A lumosacral plexus will form lacer in the hind leg buds. Sensory systems. Olfactory system. In the adult, a small area of the nasal passages is lined with olfactory epithelium that contains olfactory sensory cells whose dendrites form olfactory receptors. Axons of olfactory sensory cells comprise the olfactory nerve, cranial nervi, and synapse with cells of the olfactory bulb, functions in the sense of smell, which is located at the base of the brain. Axons from mitral cells in the olfactory bulbs form olfactory tracts that carry sensory information to ochre areas of the brain. In transverse sections of 10 mm embryos, the olfactory epithelium lines the olfactory pits, which are blind pouches in the rostral area of the head, Fig 12.15 pt. An external nares connects the olfactory pit to the amniotic cavity, Fig 12.15 r. In the adult, the external nares is an opening to each nasal passage, and the internal nares connects the back of the nasal passage to the far YNX. In the 10 mm embryo, small axonal fibers of the olfactory nerve stretch from the olfactory epithelium lining the medial wall of the olfactory pits to the rostral ventral region of RHE telencephalon wall, Fig 12.15 r. At a later stage, dendrites of the olfactory sensory cells will form and differentiate into the olfactory receptors of the nasal passages. The rostral telencephalon has not yet differentiated into the olfactory bulbs and tracts in 10 mm embryos. Visual system development of the eyes begins with the formation of the optic vasides, which are paired lateral outgrowths from the diencephalon, Figs 12.7 eh, 12.20. The cavity of the optic vesicle is the optic oval. As the optic vesicles reach the overlying ectoderm, they induce the ectoderm to thicken to form a lens placode, which later induces the optic vesicle to become a double-walled optic cup that has an inner ansari retina and outer pigmented retina, Figs 12.1 sl, 12.20. A deep fold in the ventral edge of the optic cup is the optic fissure, choroid fissure, that allows the hyaloid artery to supply the eye, Figs 12.15 m, 12.16 c, 12.20. The optic cup induces the lens placode to form a hollow LEM-VEIDC that becomes the lens of the eye, Figs 12.15m, 12.20b. In the 6 mm embryo, the lens placode has not yet developed into a lens vesicle, Fig 12.7e. The lens vesicle has formed by the 10 mm stage, Fig 12.15l, but is not as advanced as in the 72-hour chick embryo. Later, the lens vesicle will fill with lens fibers that make the lens transparent, Fig 12.20. Externally, the lens appears white, Fig 12.18. The ectoderm overlying the lens vesicle develops into the cornea, Fig 12.20. The base of the optic vesicle constricts to form the optic talk that connects the optic vesicle to the diencephalon, Figs 12.7g, 12.15m, 12.20. Fibers of the optic nerve will migrate down the optic stalk, converting it to the optic nerve, to enter the floor of the diencephalon, Fig 12.20. As optic nerve fibers cross to the other side of the midline, they form a midline thickening in the diencephalon floor called the optic chiasm, optic chiasma, Figs 12.12c, 12.20e. The outer layer of the eye, called the sclera, white of the eye, develops later from mesenchyme and neural crests surrounding the optic cup to form a tough connective tissue capsule to which the eye muscles attach, Fig 12.20. The uvca, choroid, ciliary body, iris, is the middle layer of the eye and also forms later from the surrounding mesenchyme and neural crest. The eyelids and nictitating membrane will also form later, Figs 12.18, 12.20. Auditory and vehibul all systems. The inner ear develops from the otic placode, auditory placode, that lies to each side of the myelencephalon. The otic placode invaginates to form the otic vesicle, auditory vesicle, otocyst, which is visible in 6 mm embryos, Fig 12.7 BC. By the 10 mm stage, the otic vesicle has dilated laterally to form a small duct called the endolymphatic duct. In later stage embryos, the dorsal region of the otic vesicle will form the utriculum of the inner ear that later expands to form the semicircular canals that are involved in maintaining balance. The ventral area of the otic vesicle will form the sacculus, which lies close to the auditory nerve, and the cochlea of the inner ear that is the organ of hearing. The middle ear includes the tympanic membrane, cavity, and ossicles, malleus, incus, stapes. The middle ear develops from mesenchyme surrounding the first pharyngeal pouch, and the cavity of the middle ear forms from the first pharyngeal pouch, to be discussed later. The pinna, which is the fleshy part of the external ear, develops around an opening, external auditory meatus, that connects to the inner ear, Figs 12.18, 12.25. The pinna is derived from the mandibular process of the first pharyngeal arch and the hyoid arch, second pharyngeal arch. Digestive system. Once the embryonic gut has formed, see Chapter 9, it undergoes growth, local dilations and other changes to become the adult digestive organs. At the 6 and 10 mm stages, most of the rudiments of the digestive system are present. The oral plate has already ruptured so that the cranial region of the digestive system is open to the environment. Oral cavity. The stomaticum, cranial evagination of ectoderm, and surrounding structures give rise to the oral cavity, mouth. The stomodium is continuous / with the amniotic cavity cranially, and caudally with the anterior of the pharynx, Figs 12.5b, 12.12. In transverse sections, the stomodium appears as a large cleft between the processes of the first pharyngeal arch, Figs 12, f, 12.15 lp. Teeth. See section on tooth development, p. 240. Pharynx. This is the large cavity derived from the cranial region of the foregut that lies between the stomodium and esophagus/trachea, Figs 12.12c, 12.13. The dorsal and ventral sides of the pharynx are compressed, giving the pharynx a flattened shape that is wide laterally. Because the pharynx is arched dorsally due to hection, it appears cut twice in transverse sections, Fig 12.15k. Pharyngeal arches flanking the lateral sides of the pharynx are the paired pharyngeal archi, visceral arches, bronchial arches, that are bands of mesenchyme lined with endoderm on the pharyngeal side and skin ectoderm on their external surface, Figs 12:1-5. Each pharyngeal arch has bone, muscle, nerve, cranial nerve, and blood vessel, aortic arch, components. The first pharyngeal arch is shaped like an inverted U and has two processes that are separated by the stomodium. The maxillary process is a lateral bulge that in transverse sections appears ventral to the eye and dorsal to the stomodium, Figs 12:7a. 12:15 LO. Together with the medial nasal processes, which lie between the olfactory pits, the maxillary process will give rise to the upper jaw, Fig 12.15 RS. The mandibular process is the bulge ventral to the maxillary process and stomodium that develops into the lower jaw, Figs 12.7a. 12:15 LO. The pharyngeal arches appear stacked on top of each other in transverse sections, Figs 12:7a, 12.15 kN. The second pharyngeal arch is smaller than the mandibular arch and is called the hyoid arch, Figs 12.7a, 12.150-120. The third pharyngeal arch is smaller than the hyoid arch, Figs 12.7a, 12.15 km. The fourth pharyngeal arch is less prominent than the third, Fig 12.7g. Pharyngeal grooves between the external surfaces of the pharyngeal arches are paired depressions called pharyngeal groove, Figs 12.5, 12.7a, 12.8c, 12.12, 12.15 m. Located between the mandibular process and hyoid arch, the large first pharyngeal groove is called the hyomandibular groove. The second pharyngeal groove appears between the hyoid arch and third pharyngeal arch. The third and fourth pharyngeal grooves combine to form the cervical sinus, which is located behind the third pharyngeal arch, Fig 12:15 m. PH-/re-GEAL pouches 5 paired, lateral evaginations of the pharynx form the pharyngeal pouches, visceral pouches, bronchial pouches. They line up with the pharyngeal grooves, and a closing plate usually separates them from the amniotic cavity. The FR-T pharyngeal pouches are located next to the mandibular process and are visible in 10 mm transverse sections just before the pharynx appears, Fig. 12:15 GH. In subsequent sections the pharyngeal pouches connect directly to the pharynx, Fig. 12.151J. The aconpharyngeal pouchi / lie between the hyoid and third pharyngeal arches. The third pharyngeal pouches are located in sections that contain the thyroid gland, Fig. 12.15 LM. Each fourth pharyngeal pouch is a small diverticulum from the pharynx at the level of the glottis, Fig. 12.150. The fifth pharyngeal pouch, also known as the ultima branchial body, buds off dorsolaterally from the fourth pouches, Fig. 12.1501. The derivative of the pharyngeal pouches are described in Table 9.2. Tongue rudiments of the tongue develop from swellings in the pharyngeal floor, and protrude into the pharynx, Figs 12.7e, 12.1 LC, 12.15 K. A median swelling. The tuberculum impar arises from the floor at the first. Bronchial arches. A pair of lateral lingual swellings also arise from the first pharyngeal arches and fuse in the midline. The median sulcus of the adult tongue marks their point of fusion. Another median swelling, the copula, arises from the hyoid arch and forms the root of the tongue. As the embryo develops, the tongue comes to reside in the oral cavity. Thyroid gland. A median diverticulum in the pharyngeal floor at the level of the hyoid and third pharyngeal arches differentiates into a dark, irregularly shaped rudiment of the thyroid gland, Fig. 12.7f, 12.15 LM. The thyroglossal duct which connects the gland to the pharyngeal floor has already degenerated. The thyroid is an endocrine gland, i.e., it releases its contents into the bloodstream. Esophagus. As transverse sections are traced deeper into the embryo, the pharynx appears to narrow before it splits to form the esophagus and trachea, Fig. 12.15 PR. The esophagus is the region of the foregut that is located between the pharynx and stomach. It appears circular in transverse sections, Figs 12.7 LO, 12.15 RY. The esophagus is supported by a mesentery called the mxoxophagus. Stomach. As the esophagus is traced caudally in transverse sections of 10 mm embryos, it migrates ventrally and enlarges to form the thick-walled stomach, Figs 12.7 p, 12.15 z. The lumen of the stomach appears as a slit or thin oval. The stomach has rotated approximately 45 degrees so that its dorsal side now points to the embryo's left side. The stomach is held in place dorsally by a mesentery called the dorsal mxogastrium, which will form the adult greater omentum and give rise to the spleen, Fig 12.15z. Duodenum. As the stomach is traced caudally in transverse sections of 10 mm embryos, it narrows to form the small, oval-shaped duodenum, Figs 12.7 q, 12.15 bb. The duodenum is held in place by a dorsal mesentery called the mxoduodenum, in which the dorsal pancreas will develop. The bile duct and cystic duct enter the duodenum, see liver and gallbladder. Intestines and cloaca. Continuous with the duodenum are the small intestines. They have a small lumen and a relatively thick wall. They lie adjacent to the vitelline veins and arteries. The intestines extend out into the extraembryonic chylome of the umbilical cord, forming the U-shaped intestinal loop, and the temporary umbilical hernia, also known as midgut herniation, Fig 127U. In some preparations, the intestinal loop may be difficult to find because the umbilical cord was cut too short. In transverse sections of 10 mm embryos, the darkly staining intestinal loop lies within the light pink umbilical cord, Fig 12.15 ZGG. Within the intestinal loop, the cranial limb and the caudal limbs of the intestine are visible. The cranial limb will differentiate into the jejunum and ileum of the small intestine, whereas the caudal limb will form the ascending colon and transverse colon of the large intestine. The apex of the intestinal loop connects to the yolk sac, and this point serves to separate the cranial and caudal limbs. By the 24 mm stage, the small intestine is coiled. The caudal limb of the intestinal loop is continuous with the colon that is found in more posterior sections of the 10 mm slide series, Fig. 15.1. When the colon elongates in a dorsal ventral direction, it becomes the rectum, Fig. 12.15 mm, which soon connects to the duca in more caudal sections, Fig. 12.15 ll. The cloaca is a dilation of the caudal region of the hindgut. At the 10 mm stage, the cloaca septum is beginning to divide the cloaca into a dorsal rectum, and ventral U-shaped urogenital sinus. A small region of the gut extends past the cloacal membrane as the tail gut that extends into the tail. In 6 and 10 mm pig embryos, the hindgut does not yet open to the outside, since it is covered by a Dohrnell membrane. The cloaca membrane forms where the ventral wall of the cloaca fuses with an ectodermal depression, called the proctodeum, future anus, in the ventral epidermis near the genital eminence, Figs 12.7 z, 12.15 kk. The cloaca membrane will later rupture to open the anus and urogenital sinus to the outside. Liver. A ventral diverticulum from the duodenal floor called the hepatic cli/reticulum gives rise to the liver prior to or at the 4 mm stage. In 6 to 12 mm embryos, the liver fills much of the space between the kidneys and heart, Figs. 12.3, 12.4, 12.8 to 12.11, 12.16 to 12.18. The liver is so large in part because it receives blood from much of the caudal region of the embryo and all of the extraembryonic circulation. See circulatory system. The liver functions as a temporary hematopoietic organ, produces blood cells. The liver has 4 lobes, envo-dorsal and 2 larger ventral lobes, Figs 12.7 pq, 12.15 ycc1, that are composed of strands of liver cells, hepatocytes, called hepatic cord. As the hepatic diverticulum grows, the vitelline veins interact with it to form the hepatic ethmoid, which are spaces between the hepatic cords in which blood cells are found, Fig. 5z, cc1. The mesentery called the falciform ligament attaches the liver to the ventral body wall, Fig. 1215a. The liver is attached to the stomach via the hepatogastric ligament, and to the duodenum by the hepatoduodenal ligament, Fig. 12.15 bb. Collectively the hepatogastric ligament and hepatoduodenal ligament form the lesser omentum. A small hepatic duct from the dorsal liver lobe drains bile produced by the liver into the common bile duct for storage in the gallbladder. Gallbladder. The gallbladder functions to store bile that is used to emulsify fats. The gallbladder and its duct, called the cystic duct, form as part of the hepatic diversiculum and are visible in 6 mm embryos. The gallbladder appears as a thick-walled elongated vesicle embedded in the ventral lobe of the liver, Figs 12.7r, 12.15 cc1. The cystic duct drains the gallbladder and enters the common bile duct, ductus colecticus. Pancreas. The pancreas develops from two rudiments, a dorsal pancreatic rudiment that is an outgrowth from the duodenum, and a ventral pancreatic rudiment, which is a diverticulum of the common bile duct. Both pancreatic rudiments are visible as irregular dark spots in the dorsal mesentery above the duodenum, mesoduodenum, Figs 12.7t, 12.15 cc1 dd1. The dorsal pancreatic rudiment can be traced caudally to observe it open into the duodenum by its duct called the duct of Santorini, Fig 12.15 dd1. The ventral pancreatic duct, duct of Wirsung, which connects the ventral pancreatic rudiment to the common bile duct, is degenerating and difficult to identify. These two pancreatic rudiments will later fuse to form the single pancreas of the adult that produces digestive enzymes. Respiratory system. The respiratory system develops as a diverticulum of the digestive system at the level of the caudal pharynx. The midventral diverticulum, called the laryngotracheal groove and colon, appears in embryos at the 4 mm stage and is prominent in 6 mm embryos, Fig 12.71k. In 10 mm embryos, the proximal end of the laryngotracheal groove differentiate into the larynx. The opening of the pharynx into the larynx is the V-shaped glottis. 3 swellings arise from the floor of the pharynx around the glottis, the arytenoid welling, derived from the 4th and 6th pharyngeal arches, form lateral to the glottis, and a midline swelling cranial to the glottis is the epiglottis, which is derived from the 3rd and 4th pharyngeal arches. The arytenoid swellings will form cartilages of the larynx. The epiglottis forms a temporary seal during swallowing that prevents food from entering the trachea. Distal to the larynx is the trachea / which in transverse sections appears ventral to the esophagus, Figs 12.7l, 12.15 RU. The trachea has 3 outgrowths. The most cranial evagination is the parterial bronchum, which emerges from the right side of the trachea in 10 mm m. Brius to form the upper lobe of the right lung, Fig 12.15 v1. The next 2 outgrowths appear as the trachea bifurcates, co-form 2 bronchi with lung buds, Figs 12.7n, 12.15 WLX. Later, the right lung bud will become the middle and lower lobes of the right lung. The left lung bud will generate the upper and lower lobes of the left lung. Lateral to the lungs are the pleural rhynvitides that are derived from the intraembryonic chylome and are continuous with the peritoneal cavity in early embryos, Figs 12.7 MO, 12.15 WX. Ventral to the lungs, and between the heart and liver lies the septum transversum, which is a transverse partition that separates the pericardia and peritoneal cavities. It becomes the adult diaphragm, Fig 12.15 W. Skeletal and muscular systems. The skeletal and muscular systems are derived from 3 sources of mesoderm. MCSC and CHYMC differentiates into smooth muscle of blood vessel walls. Splanchnic ectoderm generates smooth muscle layers of the gut and its derivatives. Paraxial mxid cream produces somics that differentiate into most of the skeletal muscles, Chapter 9. In 6 to 10 mm embryos, bone and muscle have not yet formed, however, somates are visible and have differentiated into 3 cell populations: 1. Dermatomte that migrates dorsally to form dermis of the skin, 2. Stratomte that migrates around the spinal cord and notochord to become cartilage of the vertebrae and ribs, and 3. Myotomte that produces muscles of the rib cage, limbs, abdominal wall, back, and tongue, Fig. L. The 6 mm pig has -36 somite pairs and the 10 mm embryo has -44 somite pairs. The area of the back in which the spinal cord is cut frontally due to flexion clearly shows the 3 somite regions in 10 mm transverse sections, Fig 12.15 UU. The adult skeletal system consists of the axial skeleton, skull, vertebral column, ribs, sternum, and appendicular. /Gluten, pectoral girdle, pelvic girdle. By the 65 mm stage, many of the developing bones are undergoing ossification, Fig 12.25. The adult vertebral column has 7 cervical, 1-4 to 15 thoracic, 6 to 7 lumbar, 4 sacral, and 20 to 23 caudal vertebrae, and 1-4 to 1-5 pairs of ribs. To form a single vertebra, the caudal half of one sclerotome fuses with the cranial half of the next caudal sclerotome on each lateral side. 2 cartilaginous scapulas form the adult pectoral girdle. Clavicles do not form in the pig, to which the forelegs are attached. The PIC girdle consists of the innominate bone that is derived from 3 bones, ilium, ischium, pubis, which are fused during feral development. The hind legs attach to the pelvic girdle. Rudiments of the limbs are the limb buds, forelimb buds, hind-learned buds. They are visible in 4.5 mm embryos and progressively lengthen as they grow, Figs 12.1, 12.4a, 12.10, 12.15y, 12.18. The apical ectodermal ridge is a thickened ectodermal structure located at the tip of each limb bud that allows the limbs to elongate, Figs 12.7s, 12.15w. The limb buds become paddle-shaped by 12 to 15 mm and eventually develop 4 toes each. The middle toes become flattened and develop hooves. The 4 limb buds are innervated by the brachia, plexus, and receive their blood supply from subclavian arteries, Figs 12.7p, 12.15xy. The sciatic plexus innervates the hind limb buds, which are supplied by the iliac arteries, a small branch of the umbilical artery at base of limb bud that is hard to see in 10 mm embryos. Urinary system. The urinary system functions to eliminate wastes. 3 pairs of kidneys develop from intermediate mesoderm, pronephros, mesonephros, and moranephros. Each kidney is associated with a duct. The small pronephros develops first. Consisting of a few pronephric tubules and the pronephric duct, the pronephros is rudimentary and does not function. It has degenerated by the 5 mm stage, however, the pronephric duct persists and is called the mesonephric duct, Wolffian duct, nephric duct, when it associates with the mesonephros. Mesonephros in 6 to 12 mm embryos, the large mesonephros occupies much of the abdomen, Figs 12.3a, 12.4b, 12. U, 12.8 to 12. 11, 12.15 mm, 12.16. Its duct is the mesonephric duct. The mesonephros is the second kidney to form and consists of many coiled mesonephric tubules and blood vessels. The medial end of each mesonephric tubule forms a double-walled cup called a glomerular capsule, Bowman's capsule, Fig 12.15a. It is associated with capillaries called glomeruli that are derived from small arteries originating from the descending aorta, Fig 12.15 gg. Glomerular capsules occupy a medial position in the mesonephros, Fig 12.15 AARR. The mesonephric tubules migrate laterally co-empty into the mesonephric duct. In transverse sections of 6 and 10 mm embryos, the mesonephric duct is easier to discern from the mesonephric tubules in more caudal sections, Fig 12-15e. When tracking the mesonephric duct caudally, it migrates ventrally and appears as a large oval at the ventral edge of the mesonephros, Fig 12.15 ll. Tracing the duct further caudally reveals that it empties into the urogenital sinus, which is a part of the cloaca, Fig 12.15 NN1. This region of the urogenital sinus becomes the urethra, drains urine from bladder. In some caudal transverse sections, the mesonephric duct is cut twice due to flexion of the embryo, Fig 12.15. The mesonephros and its duct degenerate completely in females. In males, the mesonephric ducts will persist to form efferent ductules of the reproductive system. Metanephros. The metanephros becomes the permanent kidney of the adult. In 10 mm embryos, the metanephros is just beginning to form. One way to find the metanephros in transverse sections is to locate sections in which the mesonephric duct is cut longitudinally, Fig 1-15 pp. Caudal to this area, a small ureter will be visible budding off from the caudal mesonephric duct, Fig 12.15 NN100. Following the ureter cranially shows that this duct expands into an oval surrounded by darkly staining cells. The oval is the renal pelvis, which will become the pelvis of the adult kidney. The dark cells are mtankfrognic MCSC and CHYMC that will generate the cortex of the kidney. Complex epithelial mesenchymal interactions will lead to expansion of the renal pelvis to form adult calyces and collecting tubules, and chain metanephrogenic mesenchyme generates adult secretory tubules. The blood supply of the mecanephros is via paired renal arteries, one supplying each kidney. A renal vein drains each kidney. They are visible at a later stage. During its development, the metanephros becomes bean-shaped and ascends from the pelvic area to occupy its adult position located on either side of the vertebral column, along the inner dorsal body wall at the level of the fifth lumbar vertebrae. Ascended kidneys are seen in 85 mm embryos. Urinary bladder. The urinary bladder is derived mainly from an expansion of the intraembryonic region of the allancois that lies closest to the urogenital sinus. The neck of the bladder develops from cloaca tissue. The urinary bladder has not yet formed in 10 mm embryos, but is visible at the 15 mm stage. Reproductive system. The paired gonads, ovaries, testes, are derived from intermediate mesoderm and are closely associated with the urinary system. At the 10 mm stage, the gonads are just beginning to form as paired rudiments called genital ridges that are located medial to each mesonephros. A thickening of the splanchnic mesoderm, called the germinal epithelium, marks their location, Fig 12.15 jj. At this stage, the gonads are indifferent, being neither female nor male, and have the potential to develop into either ovaries or testes. Primordial germ cells will migrate from the yolk sac through the dorsal mesentery of the gut to colonize the germinal epithelium. In addition to mesonephric ducts, Mullcrian ducts will also form later close to the mesonephric ducts in both males and females. The Mullcrian ducts are first visible in sections of 30 to 40 mm embryos. The rudiments of the external genitalia are first clearly visible in -20 mm embryos. Males. The Mullcrian ducts degenerate, however, the mesonephric ducts differentiate into the epididymis, vas deferens, seminal vesicles, and ejaculatory ducts. The epididymis is also derived from mesonephric tubules. The prostate gland forms from the epithelium surrounding the urethra. Once formed, the testes and associated structures descend into the scrotum, Chapter 3. Male reproductive structures are not visible in 10 mm embryos. Females. The mesonephric ducts degenerate, however, the paired Mullcrian ducts persist. Cranially, the Mullcrian ducts form the two separate uterine horns, cornua, and oviducts, which become closely associated with the ovaries. Caudally, the Mullcrian ducts fuse at their cloaca end to form the uterine body and vagina. The vagina and urethra both open into the urogenital sinus, vestibule, in adult sows. Circulatory system. Chapter 9 introduced general features of cardiogenesis, vasculogenesis, and the embryonic circulatory system. Thus, familiarity with this chapter is recommended before proceeding. For simplicity, paired structures are described as single. In 4 to 10 mm embryos, the embryonic circulatory system is functioning, but does not yet resemble the adult system. Overview of embryonic circulation. There are 3 major circulatory arcs in pig embryos, with the heart as the pump at the center of each. 1. The allantoic circulation carries blood to and from the placenta through umbilical blood vessels, allantoic blood vessels. 2. The Vicklin circulation carries blood to and from the yolk sac through Vick-linked blood vessels, omphallomesenteric blood vessels. These 2 circulatory arcs are considered extraembryonic circulation because they lie mostly outside the early embryo. The intraembryonic circulation is confined to the body of the embryo and functions to bring nutrients and oxygen that were collected by the other circulatory arcs to the embryo, and carry wastes from the embryo to either the allantoic circulation or the mesonephros. Heart. The heart is located in the pericardia. CNITY, which is a part of the intraembryonic chylome, Fig 12-150Y. Later the heart will be surrounded by the prenerdium, a thin-walled sac. Early during development, the heart is a hollow tube that has a single atrium and ventricle connected to each other via the single atrioventricular canal, Chapter 9. At 6 to 10 mm stages, the muscular heart consists of 4 chambers that include the left and right atria and left and right ventricles, Figs 12.71Q, 1215QZ. The atria are separated by the double-walled incratrialptum, Figs 12.7L, 12.155, except for a small opening, called the foramen ovale, that allows blood to move from the right to left atrium until the foramen ovale closes at birth. The intraventricular septum incompletely separates the left and right ventricles at these stages, Figs 12.71-12.15 UX. The temporary internal opening between the ventricles is the interventricular foramen, Figs 12.7L. 5 UV. Externally, a small depression on the ventral side of the heart called the intraventricular yaku marks the boundary between the ventricles, Figs 12, kp. 12.15 UX. The right atrium is directly connected to the inmagna, which receives blood from the 1. Common cardinal veins, 2. Umbilical veins, and 3. Vitelline veins, Figs 12. M0, 12.15 UW. The sinoatrial valve, valvuli venosi, has right and left valve flaps that prevent blood entering the right atrium from flowing back into the sinus venosus, Fig 12.15 U. As the heart grows, the sinus venosus is absorbed into the right atrial wall to become the sinoatrial node, a parch of Purkinje fibers, which is the pacemaker of the adult heart. The right atrium is a large chamber of the heart that is dorsal to the right ventricle, Figs 1271N, 1215QW. It is connected to the right ventricle via the right atrium ventricular canal, Figs 12.7L, 1215 UV. The endocardial cushions are 2 masses of lightly staining mesenchymal tissue that grow together around the atrioventricular canals. RO later form the heart valves, and to close the interventricular foramen. The tricuspid valve has 3 flaps, develops within the right atrioventricular canal, and the bicuspid valve, mitral valve, has 2 flaps, forms within the left atrioventricular canal. In transverse sections, the thick-walled right ventricle appears to lie ventral to the right atrium, Figs 12.7 kp. 1215 RY. It is continuous not only with the right atrium, but also the cona truncus, a region of the heart that becomes subdivided into the pulmonary trunk and aortic trunk, ascending aorta, Fig 1215 Q. The cona truncus has also been called the conus arteriosus, bulbus cordis, conus, aortic sac, and truncus arteriosus. In the 6 mm embryo, the cona truncus is a single trunk, but by the 10 mm stage, the hulnar ridges of the conotruncus have grown out into the lumen, internal space, of the conotruncus and fused to form the bulbarptum that separates the pulmonary and aortic trunks, Figs 1 2.7 Hi, 1215 Q. The left atrium is a large chamber dorsal to the left ventricle. It receives a single pulmonary vein that will later branch into 4 pulmonary veins. The left atrioventricular anal connects the left atrium to the left ventricle, Figs 1271N, 1215 QV. The thick-walled left ventricle lies ventral to the left atrium, Figs 12.7 KO, 12.15 SZ. Blood leaves the left ventricle through the aortic trunk. The walls of the adult heart have 3 layers, pericardium, visceral pericardium, which is the outer layer lying closest to the pericardia. Cavity, myocardium. Thick muscular middle layer, endocardium, which forms the inner layer that is in contact with blood cells. In 6 and 10 mm embryos, the walls of the heart are formed of 2 layers. 1. The endocardium is the internal lining of the embryonic heart, and 2. The epimyocardium, which will later differentiate into the myocardium and epicardium. The ventricular epimyocardium is characterized by trabeculae carniae, which are irregular dense bands of muscle cells that project into the lumen of the ventricle, Figs 1-2. KO, 12.15 UY. Blood enters the embryonic heart through the sinus venosus, moves to the atria, and passes into the ventricles, where it is pumped out the conotruncus for distribution to the rest of the body. LNTRAF. Embryonic blood vessels. Blood that is destined for circulation within the embryo leaves the aortic trunk of the conotruncus, passes through the ventral aorta through the aortic arches and into the dorsal aorta, where it is distributed to the body. The cranial cardinal veins return blood from the cephalic region of the body, and the caudal cardinal YSTCM drains blood from the caudal part of the body. Near the heart, the cranial and caudal cardinal veins fuse together to form a single common cardinal vein which drains into the sinus venosus. Blood from the intraembryonic and extraembryonic circulation mixes in the heart, allowing exchange of nutrients, wastes, and gases. Specific intraembryonic blood vessels that are visible in 6 and/or 10 mm embryos are described next. Arteries. Arteries are blood vessels that carry blood away from the heart, under pressure from the heart. They usually have thicker walls than veins. They begin at the heart and end in capillaries, smallest blood vessels, of the tissues/organs. Aortic arches. 6 pairs of aortic arches develop to each side of the pharynx, Figs 9.16 to 9.17. They extend from the ventral aorta to the dorsal aorta. Not all the aortic arch pairs are present at the same time. In 10 mm embryos, the first, second, and fifth aortic arches have degenerated, and the third, fourth, and sixth aortic arches are functional. The large third aortic arches extend from the ventral aorta, Fig 12.15N, and traverse the third pharyngeal arch, Fig 12.15 IM, before joining the dorsal aorta, Fig 12.15H. The third aortic arches will become the common carotid arteries and the proximal part of the internal carotid artery. The distal region of the internal carotid arteries is derived from the dorsal aorta. Often in the same transverse section, the fourth aortic arches are visible leaving the ventral aorta and joining the dorsal aorta, forming a large U-shaped structure with the arms of the U being formed by the fourth aortic arches, and the base of the U is the ventral aorta, Fig 12.15N1. The right fourth aortic arch will become the right subclavian artery, supplies the right foreleg, whereas the left fourth aortic arch will become the arch of the aorta, Fig 9.17. The sixth aortic arches are continuous with the pulmonary trunk and connect to the dorsal aorta, Fig 12.15Q. While the distal end of the right sixth aortic arch degenerates, the distal left sixth aortic arch becomes the ductum arteriosus, an opening between the left pulmonary artery and arch of the aorta that persists until birth to allow blood to bypass the functionless lungs. After birth, the ductus arteriosus closes and becomes a ligament called the ligamentum arteriosum. Dorsal aorta and descending aorta. The dorsal aorta is the main vessel that distributes blood to the intraembryonic circulation, Figs 12-7 DQ, 12.15 GW. In the head, the dorsal aorta is paired and extends forward as the internal carotid artery. Below the region of the eighth somoid, the dorsal aorta has fused to form a single blood vessel called the descending aorta, Figs 12,7 RZ. 12.15 WLSS, it will become the adult aorta, also known as abdominal aorta. Caudally, the descending aorta NAR rose and extends into the tail as the median, caudal artery, Fig 12.11c. The many arteries that branch, dorsally, laterally, and ventrally, from the dorsal aorta and descending aorta will be described below. Internal, external, and common carotid arteries. The region of the ventral aorta that gives rise to the third aortic arch becomes the common carotid artery. The internal carotid artery is derived from the third aortic arch proximally, and distally is an extension of the dorsal aori that supply the brain, Figs 12-7 DF, 12.15 DJ. The region of the ventral aorta that gave rise to the first and second aortic arches forms the external carotid artery that will later generate thyroid, lingual, and maxillary arteries. Basilar artery. The unpaired basilar artery is an extension of the internal carotid arteries that have turned dorsally at the mesencephalon to form the posterior communicating arteries and fused at the level of the mesencephalon to form the basilar artery. It lies in the midline ventral to the rhombencephalon, Fig 12.1 SEI. Intersegmental arteries. These small, paired arteries are dorsolateral branches of the dorsal aorta that extend between the somates. They are easier to identify in 10 mm transverse sections at the level where the lower back is curved frontally. They appear as dark horizontal streaks between the somates, Fig 12.151t. Subdavian arteries. The seventh intersegmental arteries, which are dorsolateral branches of the dorsal aorta, enlarge to form the left and right subclavian arteries. They will supply the forelegs. In transverse sections of 10 mm embryos, they appear as a branch extending from the descending aorta into the foreleg buds, Fig 12-15x. Pulmonary arteries. The pulmonary arteries arise caudally from the sixth aortic arches and extend to the lung buds. Unlike most mammals in which the pulmonary arteries are paired, pig pulmonary arteries fuse proximally to form a short, median vessel that extends toward the trachea. Distally, this vessel branches to form the right and left pulmonary arteries that supply the lungs, Fig 12.15 RT. Superior mesenteric and celiac arteries. See vitelline arteries under extraembryonic blood vessels. Mesonephric arteries. These are lateral branches of the descending aorta that extend into the mesonephros to supply the mesonephric glomeruli, Figs 12.7t, 12.15 GG. Renal arteries will supply the metanephric kidneys. External and internal iliac arteries. The paired common iliac arteries branch laterally from the umbilical arteries at the level of the hind leg buds, Fig. 12.LSNN artery not shown. The short common iliac arteries give rise to the 1. External ilia. Arteries that branch to form the femoral arteries that supply the hind legs, and 2. Internal iliac arteries also called hypogastric arteries, proximal region of umbilical arteries, that supply the pelvic region. These blood vessels are difficult to identify in 10 mm embryos. Caudal artery. A median blood vessel of the tail that is the caudal extension of the descending aorta, Fig 12-15 HH. Veins. Veins are blood vessels that drain blood from the organs and return it to the heart under relatively low pressure. They usually have thinner walls than arteries. They generally begin with capillaries and end in the great vessels that return blood to the heart. Cranial cardinal veins. These paired veins, also called anterior cardinal veins, precardinal veins, bring blood from the head and neck into the common cardinal veins, Figs 12-7 BK, 12-15 F1. In Transverse sections, the cranial cardinal veins are cut twice due to hection, Figs 12.7 df, 12.15 fm. They appear as long vessels when cut frontally, Figs 12.7 c, 1-15a. They lie dorsal to the petrosal and nodose ganglia, Fig 12.15 gl, and lateral to the dorsal aorta, Fig 12.15-0, until they move ventrally to join the common cardinal lines, Fig 12.15 ps. The cranial cardinal veins become the adult internal jugular veins. Common cardinal veins. Also called the ducts of Cuvier, these veins receive blood from the cranial and caudal cardinal veins, and empty into the sinus venosus. They lie lateral to the dorsal aorta, and then migrate ventrally to join the sinus venosus, Figs 12.7 ko, 12. 15 rv. The right common cardinal line together with the right cranial cardinal vein become the adult superior vena cava. Caudal cardinal veins. These veins, also called postcardinal veins, drain the caudal region of the embryo and empty into the common cardinal veins. They lie dorsolateral to the descending aorta, Figs 12.7 mt, 12.15 wx. At the level of the kidney, they are interrupted by mesonephric tubules and become difficult to identify, Fig 12.15 yff. The left caudal cardinal vein will later degenerate, whereas the right will form the adult aegus vein. Subcardinal veins. Lying parallel to the caudal cardinal veins in the trunk of the embryo and medial to the mesonephros, the subcardinal veins fuse with each other in several places to form many anastomoses at the level of the mesonephros, Fig 12.15 EENN. They also anastomose with the caudal cardinal veins. They drain the medial mesonephros and empty into the inferior vena cava. Pulmonary veins. These veins carry blood from the lungs directly into the left atrium. In the 10 mm embryo, a single pulmonary vein appears as a small diverticulum from the left atrium at the level where the left common cardinal vein lies next to the lateral atrial wall, Fig 12.15 UV. Later this vessel branches to form 4 pulmonary veins. Inferior vena cava. This vein is derived from several blood vessels including the right vitelline vein, right subcardinal vein, right supracardinal vein, and right caudal cardinal vein. It can be identified between the liver and heart, Fig 12.15 WDD. It receives blood from the ductus venosus and empties into the sinus venosus. It is also called the postcaval vein and posterior vena cava. In the adult, it brings most of the caudal venous blood to the right atrium of the heart. Ductus venosus. Region of umbilical vein that has entered the liver. It carries blood from the vitelline veins and left umbilical vein into the inferior vena cava, Fig 12.15 YBB. Hepatic veins. These are paired veins that are derived from the left and right vitelline veins, ompulomesenteric veins. They drain the cranial liver before emptying into the inferior vena cava.
Hepatic portal vein.
This blood vessel is derived from portions of the right and left vitelline veins. It spirals around the gut, and carries blood from the superior mesenteric vein to the ductus venosus and sinusoids of the caudal liver. In transverse sections of 10 mm embryos, it can be traced caudally from the right dorsal lobe of the liver to the dorsal mesentery, where it lies to the right of the duodenum, Fig 12.15 bbcc. It then becomes located near the dorsal and ventral. Pancreatic rudiments, Fig 12.15 dd1, and lies to the right of the small intestine, Fig 12.15 ee. The hepatic portal vein runs along the intestinal loop into the umbilical cord, where it will later split into LE and right vitelline veins that drain the yolk sac. Superior mesenteric vein. The superior mesenteric vein is derived from the proximal part of the left vitelline vein. It lies in the dorsal mesentery to the left of the duodenum, Fig 12.15 ddjj. It extends into the umbilical cord to drain the intestinal loop, Fig 12.15 hh1. Intersegmental veins. These veins lie cranial to the intersegmental arteries. They drain the somates and empty into the cardinal venous system. Subclavian veins. Capillaries of the forelimb buds are drained by subclavian veins that empty into the cranial cardinal veins, Fig 12-7p. They are the seventh intersegmental veins. Iliac veins. Capillaries of the hind limb buds are drained by the paired iliac veins that empty into the caudal cardinal veins. They are not always visible in 10 mm embryos. Caudal vein. This vein drains the tail blood into the caudal cardinal veins, Fig 12.15hh. Extraembryonic blood vessels. The extraembryonic blood vessels include the vitelline, omphilomesenteric, blood vessels and umbilical, allantoic, blood vessels. They function to carry nutrients and oxygen into the embryo, and wastes from the embryo. Vejatu.ine are terges. They are branches of the dorsal aorta that fuse to form a single, ventral median artery called the superior mesenteric artery, common vitelline artery, that lies in the dorsal mesentery of the embryo, Figs 12-7 SV. 12.15 ccjj. This artery supplies the intestinal loop, small intestine, and yolk sac. Tracing the artery in transverse sections shows that the distal end is in the intestinal loop near the yolk stalk, Figs 12.7s, 12.15z, and the proximal end emerges from the descending aorta, Figs 12.7v, 12.15. This is the first of three major enteric blood vessels to form. The other two blood vessels, the celiac artery and inferior mesenteric artery, form later as branches originating from the dorsal aorta that supply the stomach and large intestine, respectively. Vitl.I.ine veins and common VLTELLINE vein. The right and left vitelline veins function to drain the yolk sac and intestines. In 6 mm embryos, they appear near the yolk sac and enter the liver, where they form many anastomoses, Fig 12. Pu. In 20 mm embryos, opposite portions of the right and left vitelline veins degenerate while others persist to form the hepatic portal vein. See also superior mesenteric vein and hepatic portal vein. UMBLLJCAL arteries, paired lateral branches of the descending aorta at the level of the common iliac arteries at the hind limb buds. They are the largest branches of the descending aorta. The umbilical arteries migrate along the allantoic stalk and extend into the umbilical cord. In transverse sections, they lie to each side of the allantoic stalk, Figs 1-2. 7vx, 12.152rr, 12. 19, they carry blood from the embryo to the allantois/placenta. Umbilical veins, paired blood vessels that carry oxygenated blood from the placenta to the embryo's liver, Figs 12.7rz, 12.15 bboo. In 6 to 10 mm embryos, the right umbilical vein is degenerating and thus often appears smaller in transverse sections, Figs 12.7s, 12.15 ee. The left vein enlarges and enters the liver as the ductus venosus before terminating in the inferior vena cava, Fig 12.15 ybb. Due to flexion, the umbilical veins are cut twice, Fig 12.15 gg. In the umbilical cord of a late-stage fetus, the single left umbilical vein is present along with the two umbilical arteries and allantoic stalk, Fig 12.19. The umbilical vein has a thinner wall than the umbilical arteries. Tooth development. Teeth begin their development under the oral epithelium, oral ectoderm, and later erupt to the surface in the oral cavity. The crown, area above the gums or gingivae, forms first, and then the roots, area below the gum line. Forming last are the cementum, a bone-like substance that covers the roots, and the periodontal ligament, periodontal membrane, a fibrous structure that connects the cementum to the bone of the socket, alveolus, that holds the tooth. Pigs are omnivores, diet has both plant and animal material, and their teeth reflect this. Adults have 3 incisors, 4 cutting, 1 canine, used for puncturing and holding, 4 premolas, for grinding and crushing, and 3 molars. For grinding and crushing, on each side of the upper and lower jaws. The first teeth to erupt are the 2 canines and the third pair of the incisors. At birth, the neonatal piglet is missing its molars. They will erupt later. 2 generations of teeth form, the deciduous primary teeth, milk teeth and the permanent teeth. Their pattern of formation is very similar. The adult tooth has 2 hard extracellular matrix components that surround a central pulp of connective tissue. 1. An inner core of calcified dentine, originates from mesenchyme, that is surrounded by 2, a cap of hard enamel derived from ectoderm. The pulp cavity of the tooth is filled with loose connective tissue called dental pulp. Teeth form through reciprocal interactions between the mesenchyme and overlying oral epithelium. First, the mesenchyme induces the epithelium to thicken into a placode that secretes substances that then induce the mesenchyme to change. The modified mesenchyme then influences the altered epithelium to change further, and this process continues until a tooth is formed. An important step in tooth development is migration of neural crest cells into the upper and lower jaws. The neural crest cells aggregate to produce mesenchyme that underlies the oral epithelium at specific sites. These sites become areas where teeth form. During tooth morphogenesis, oral epithelium thickens to form a placode or dental lamina, labiodental lamina, that invaginates, pushing into the underlying mesenchyme to form the dental ledge of a tooth bud, Fig 12.21ab. The neural crest-derived mesenchyme condenses around each tooth primordium. The dental lamina dilates to form an enamel organ that pushes further down into the gums mesenchyme. Condensed mesenchyme deep to the enamel organ forms the dental papilla. The dental papilla pushes up into the enamel organ, causing the enamel organ to become shaped like a cap, Fig 12.22a, and then a bell, Figs 12.23, 12.24, as it grows around the dental papilla. At the cap stage, the concave border of the enamel organ, LIES against the dental papilla, differentiates into amyloblasts, enamel-producing cells, Fig 12.22. At the bell stage, the surface of the dental papilla becomes covered with odontolasts, cells derived from neural crest that secrete dentine, Fig 12.23. At the late bell stage, amyloblasts and odontoblasts begin secreting precursors of enamel and dentine. This begins at the tip, apex, of the tooth and continues down the sides, Figs 12.23, 12.24. The amyloblasts and odontoblasts eventually become separated by the enamel and dentine layers that they secrete, Fig 12.24c. Connective tissue surrounding the enamel organ becomes the dental follicle that differentiates into the periodontal ligament and cementum of the root of the tooth. Integument. The integument is composed of the epidermis on the body surface, and the dermis lying immediately under. The epidermis, Fig 12.24a. Between the integument and deep body musculature is the hypodermis, superficial fascia, subcutaneous fat, which is loose connective tissue and adipose tissue. The epidermis is derived from the ectoderm, and the dermis is derived from mesoderm, dermatome, somatic mesoderm, and neural crest. Later, the skin will specialize to form hair, hooves, and glands. Having studied organogenesis in several model organisms in previous chapter, the developmental biology student is poised to apply this knowledge to the human embryo. As new discoveries in model organisms provide key insights into the fundamental regulatory genes of development, the need to fully understand the anatomy of human embryos and function of human genes is even more pressing. How disruption of the function of these genes relates to human congenital abnormalities is a major focus of research today. For example, mutations in the KIT gene lead to piebaldism. Other examples include SHH, craniofacial and limb abnormalities, FGFR3, thanatophoric dysplasia and Ocker dwarfisms, PAX, congenital megacolon or Hirschsprung disease, HOXD13, syndactyly, CBFL, cleidocranial dysplasia, and SOX9, campomelic dysplasia. Understanding the generic causes of these congenital malformations will not only lead to correct diagnosis, but also safe and effective therapies. Source of human embryos. Unlike embryos of animal models which developmental biologists can collect in a scientifically controlled laboratory environment, human embryos become available haphazardly, such as when a pregnancy is terminated by natural causes, by accident, or deliberately, as in therapeutic abortions. A question often asked is, where do the human specimens of museum collections come from? Up to -1950, pregnancy tests did not yet exist. Patients who had surgery for a hysterectomy, removal of the uterus, became a source of embryos when the surgeon unknowingly terminated the pregnancy upon removal of the womb. With advances in modern pregnancy testing, surgeons can avoid performing a hysterectomy on a pregnant woman, which is considered unethical today. Another source of embryos can be a patient who had a therapeutic abortion, such as in the case of a tubal pregnancy, ectopic pregnancy, in which the embryo has implanted in the fallopian tube instead of the uterus. Other embryos came from spontaneous abortions, loss of embryo/fetus due to natural causes, or accidents. Often embryos of spontaneous abortions are severely abnormal and cannot be used to draw conclusions about normal development. The embryos shown in this chapter are from the Carnegie collection and were obtained between 1914 and 1947. Each embryo in the collection was assigned a number, and will be referenced by their Carnegie collection number. Establishing embryonic age. To get a better sense of the events of human development in utero, within the uterus, the entire process has been divided into phases. Obstetricians often divide the entire 9-month period of human development into 3 trimester, each 3 calendar months long. Developmental biologists divide human development in utero into 2 stages, the embryonic period, embryogenesis, which begins at fertilization and concludes at the end of 8 postovulatory weeks, and the fetal period, which begins with postovulatory week 9 and ends at birth. Primordia of all the tissues and organs, and the embryonic axis are formed during the embryonic period. During the fetal period, the tissue and organ primordia further differentiate, grow, and begin to function. Thus the fetal period is characterized by the rapid growth of the fetus, increase in size and body weight, and onset of organ function. The technique of ultrasonography, in which sound waves with frequencies beyond the range of human hearing are used to obtain living embryo/fetal morphology in utero, has contributed greatly to our understanding of human development and the ability to determine the age of the embryo/fetus. During the first trimester, ultrasonography is used to determine embryonic fetal age. During the second trimester, it can be used to detect fetal abnormalities. Fetal growth, placenta location, and complications due to fetal movements can be determined using ultrasonography during the 3rd trimester. Since the exact date of fertilization may be unclear, and often the only available date is the first day of the last menstrual period, LMP, a gestational age, also called LMP age or menstrual age, can be assigned to the embryo. LMP age begins at time 0, 14 days before fertilization, and ends at birth, week 40, 9.2 months, 280 days. Note this is 2 weeks ahead of the true age, age from fertilization, post-ovulatory age, of the embryo. Normal gestation, pregnancy, averages 38 weeks in posovulatory age and 40 weeks in LMP age. Because it is difficult to estimate human prenatal age, several staging series have been developed to describe development during the embryonic period. This chapter uses the Carnegie system of staging, which was developed by George Streeter and modified by Ronan O'Rahilly and Fabiola Muller using specimens from the Carnegie collection. In this classification system, the embryonic period is divided into 23 Carnegie stages. The first stage begins with fertilization, and the series ends with stage 23, which is the end of the embryonic period. Each Carnegie stage indicates the embryo's age in days post-ovulation, length of time since the last ovulation, and provides the length of the embryo in addition to details on external and internal features. The crown rump length, CR length, from crown of head to buttock region of embryo, and number of somates are useful external landmarks to help determine the extent of development of human embryos. The following additional measurements have been used to help determine embryonid fetal age: CR length, crown heel length, CH length, from crown of head to heel of fetus, foot length, length of ossified femur, circumference of the head, circumference of the abdomen, and body weight. Developmental events during the first 11 stages of the embryonic period have been addressed in previous chapters. NASA's ZADIS 13 focuses on human organogenesis from Carnegie stages 12 to 23 to gain better insight into the remarkable journey all of us have taken. The 2D of the human embryo provides a deeper understanding of the relationship of structures and organs in fetal and adult anatomy. This is readily seen in transverse sections at stages 12 and 13, Figs 13.3, 13.8. These transverse sections highlight similarities of human embryo morphology to the 10 mm frog embryo, 72-hour and 96-hour chick embryos, and 6 mm and 10 mm pig embryos. Because of the similarities of pig and human embryos, most of the detailed discussion of the pig embryo in Chapter 12 is directly applicable to the human embryo. Therefore, Chapter 13 will only briefly describe human development during the embryonic period. Embryonic period. Stage 12. The stage 12 embryo, days 26, 27, is characterized by a large head and smaller, elongated body, Fig 13.1. Embryo size ranges from 3 to 5 mm. Flexion causes the embryo to become curved like the letter C, and many embryos at this stage also exhibit torsion in the lower half of the body. The otic vesicles have formed, Fig 13.3a. The optic vesicles are evident, Fig 13.3b. The brain has already differentiated into prosencephalon, mesencephalon, and rhombencephalon, Figs 13.1, 13.3. The cranial flexure is visible as a ventral bend in the brain at the level of the mesencephalon. The caudal neuropore closes by the end of this stage. 3 pairs of pharyngeal arches are present, Fig 13.1. The heart appears as a bulge close to the head and the embryo is becoming vascularized, Figs 13.1, 13.3. The upper limb buds, future arms, are beginning to develop, Figs 13.1, 13.3c. Between 21 and 29 somite pairs lie outside the lateral sides of the neural tube, Fig 13.3. The gut has differentiated into foregut, midgut, and hindgut derivatives, Fig 13.3c. Primordia of the respiratory tract and liver are present, Fig 13.3b, c. A caudal prominence, tail, is present. The stage 12 embryo, Carnegie embryo number 5923, shown in figures 13.1 to 13.3, was fixed in formol in 1929, embedded in paraffin, and serially sectioned at 10 m, transverse to the long axis of the embryo. The embryo was subsequently stained with alum cochineal. After fixation, but before sectioning, the embryo was 4 mm in length. It has 28 somite pairs. Stage 13. The stage 13 embryo, days 28 to 31, week 4, is C-shaped due to flexion, Fig 13.4. It is approximately 4 to 6 mm in CR length. The head is large and 4 pharyngeal arches are evident. The otic vesicle is closed and has detached from the skin ectoderm, Figs 13.4, 13.5, 13.8. The embryo has -30 somite pairs. The heart forms a large prominent bulge under the head, Figs 13.4 to 13.7, 13.8 nt. This bulge is more prominent than that of the pig or chick embryo. The lower limb buds, leg buds, are visible, Fig 13.4. Primitive blood cells are circulating in the embryo, and gaseous exchange is taking place with the maternal blood in the intervillous spaces, Figs 13.5, 13.6. Transverse sections show the lung buds and trachea rudiments, Fig 13.80. The mesonephros containing mesonephric tubules is beginning to differentiate and is very small compared to that of the pig, Fig 13.8x. Germ cells are migrating. The umbilical cord is present, Fig 13.4, 13.8 ux. The stage 13 embryo, Carnegie embryo number 836, shown in figure 13.8 was fixed in 1914 in saturated aqueous mercuric chloride containing 5% glacial acetic acid. After fission its CR length was 4 mm. It was embedded in paraffin, and serially transversely sectioned at 15 m. The 247 transverse sections that were generated were mounted on 14 large glass slides, and stained with carmine, alum cochineal. The embryo has 32 somite pairs along the body axis. STCGE14. The stage 14 embryo, day 32, week 5, is characterized by a 5 to 7 mm CR length, Fig 13.9. The optic cups are forming, and the lens has a pit open externally. The nasal plate is flat. The growth of the second pharyngeal arch over the third pharyngeal arch has formed a depression, the cervical sinus. The mesenteric ridge reveals the mesonephros, as it extends into the abdomen. The metanephros is just beginning to form as the ureteric bud acquires a cap of metanephrogenetic mesenchyme. Primordial germ cells have reached the gonadal ridge, genital ridge. The upper limb buds appear paddle-shaped and are elongating. Stage 15. A 7 to 9 mm CR length characterizes the stage 15 embryo, days 33 to 36, Figs 13.10 13.12. The pontine flexure is evident in cleared specimens. Axons of the olfactory neurons reach the telencephalon. The cerebral vesicles are evident. The nasal pits are forming. The lens vesicles are closed. The hand PHLTC is visible at the distal end of the arm. The shoulder and arm regions are evident. The midgut is beginning to elongate and form a loop. The ureteric bud is expanding to form the renal pelvis. Large numbers of germ cells populate the gonadal ridges. The genital tubercle is visible lying between the umbilical cord and the cloacal membrane. At the end of this stage, the lower limb bud is paddle-shaped. Stage 16. Stage 16 begins at 37 days and ends at 40 days, week 6, Fig 13.13. Stage 16 embryos have 8 to 11 mm CR length. The head is very large. Pigment in the retina is visible as a brown ring in the eye. The optic fissure is present in the optic cup. The upper lip and nose are developing. The nasal cavity is connected to the oral cavity. Small elevations of the first and second pharyngeal arches form the auricular hillox, future auricle or pinna of external ear, that surround and opening the external auditory gatus, first pharyngeal groove. The gut is beginning to rotate. The liver forms a prominence in addition to the heart. The gonads are differentiating. The mammary ridge appears. The foot plate is visible at the distal end of the leg bud. The thigh and leg regions are also evident. The caudal eminence begins to recede. Stage 17. An 11 to 14 mm CR length characterizes the stage 17 embryo, days 41-43, Fig 13.14. The head is very large and the cervical flexure has bent it sharply over the heart and liver prominences. The trunk is slightly straighter. The auricular hillocks are prominent on the mandibular and hyoid arches. The eye appears as a brown ring due to C retinal pigment. The cavity of the lens vesicle is reduced in size. The facial features and palate are forming. Chondrification of the limb bones and some vertebrae is beginning. The vertebral column has 36 skeletal, cartilaginous, elements. The lungs are lobed. The mesonephros is functional. Stage 18. The stage 1-8 embryo is 44 to 47 days old, week 7, Fig 13.15. The CR length is 13 to 17 millimeters. The trunk of the body is relatively straight, while the cervical flexure has bent the head over the large heart prominence. The eyes are obvious and appear dark with pigment. The eyelids are starting to form. The cavity of the lens vesicle is reduced to a slit. The auricles are visible. Digital rays are present in the large hand plates. The beginnings of grooves between the distal ends of the digital rays reveal the future location of the fingers. Elbows are starting to form. The rapidly growing intestines spill into the umbilical cord, forming the temporary umbilical hernia. The lumbar flexure has curved the lower region ventrally. Collecting tubules and nephrons are differentiating in the metanephros, but glomeruli have not yet formed. In males, testicular sex cords are present. Stage 19. The stage 19 embryo is 47 to 49 days old, week 7, and its CR length is 17 to 20 millimeters, Fig 13.16. The head is no longer at a right angle to the trunk. The trunk of the body elongates and straightens. The arms have elbows that appear to curve over the heart prominence. The lower limb bud differentiates into thigh, leg, and foot. Toe rays are developing, but interdigital notches have not yet formed. The caudal eminence has almost completely receded. Ossification may begin. Stage 20. Stage 20 embryos are 50 to 51 days old, week 8, and have a CR length of 21 to 23 millimeters, Fig 13.17. The cerebral hemispheres have grown to cover at least 2/3 of the diencephalon. The arms are bent at the elbows and the webbed fingers curve over the heart prominence. The metanephros has S-shaped renal tubules. Stage 21. A CR length of 22 to 24 millimeters characterizes stage 21 embryos, days 52 to 53, Fig 13.18. The cervical flexure is reduced. The forehead appears large. The pigmented retina makes the eyes dark. The elongating fingers are separated and the toes are beginning to separate. The hands and feet are close to the embryo's midline. Glomerular capsules are forming in the metanephros. A stubby caudal eminence is still present. Stage 22. The stage 22 embryo is 54 to 55 days old, week 8, Fig 13.19. It has a CR length of 25 to 27 millimeters. The eyelids are more developed. The external ear appears more like the adult form. The fingers of one hand can overlap the other. The legs are elongating. Individual toes are visible. The metanephros has several large glomeruli. Stage 23. Stage 23 embryos are 56 to 57 days old, week 8, and have a 28 to 32 millimeters CR length, Fig 13.20. Reduction of the cervical flexure makes the head more erect. The rounded head occupies almost half the length of the embryo. The face appears more like the adult, with the eyes forward rather than at the side of the head. Eyelids are open. The auricle of the ear is evident. A distinct neck is visible. The limbs are longer and more developed. The umbilical hernia is still present. The external genitalia are well developed, but it is usually too early to determine the gender of the embryo by this means only. The caudal eminence has receded.